Hi, everybody. Welcome to Chapter 9. So, in this chapter, we're going to be looking at biotechnology. So, biotechnology is the use of both microbiological and biological techniques to answer different questions and to help make the world a better place, basically. So, let's look at a glimpse of our history. In the 1970s, late 70s to early 80s in Argentina, there was a war in which there were military junta who killed lots of citizens, so thousands of, in general, just regular people, anybody who didn't agree with the junta. There were about 200 children that survived these killings. And so they weren't killed. Instead, they were kidnapped and they were placed in pro-junta families. Dr. King from UC Berkeley, once the war ended and a democratic government took over, was tasked with the job of reuniting children with relatives. So she used biotechnology to analyze DNA sequences, and she was able to look at the mitochondrial DNA. She couldn't look at the parents' DNA because the parents were dead or missing, but she was able to use the mitochondrial DNA and was able to match children with their siblings as well as with maternal grandparents or maternal family members, I should say. So what is biotechnology then? I just said this before, but the use of microbiological and biochemical techniques to solve different practical problems. So what are some of these problems? How do we make an organism have unique characteristics? How do biologists amplify DNA so they can run more tests? How can biologists isolate organisms with new genes? How can we catch a criminal? How can we identify specific mutations? How can we determine parentage of a child? So for biotechnology, there are multiple applications that we can use biotechnology on. So genetic engineering, where we are able to modify organisms to make them more valuable because they contain medicinal qualities, or they're able to grow in environments where these organisms normally couldn't grow. We can sequence DNA so that we can identify lineages from organisms, and we can kind of find ancestry, as well as we can find mutations within DNA that might code for certain disorders. Polymerase chain reaction, we can amplify DNA so that we can run more tests. This is very useful in trying to identify paternity or identify maternity if that was a problem, or using for DNA fingerprinting. And then probe technologies, where we can add a fluorescent probe to DNA to study gene expression, specifically in the medical world, to find out what genes are turned on in cancer patients, or if you have a specific gene for a certain disorder. So we're going to look at restriction enzymes first. Restriction enzymes are basically biological scissors. And what they do is they cut up DNA that has a certain nucleotide sequence. The sequences are typically palindromic, and palindromic means it's going to be read the same way forward and backward. And so these restriction enzymes recognize these palindromes, and then they chop the DNA at specific regions within that palindromic section of DNA. Restriction enzymes don't care what DNA they're cutting up. They will cut up bacterial DNA, they will cut up viral DNA, they will cut up animal DNA. They don't care. And a lot of times when they cut, they produce restriction fragments that have sticky ends. And sticky ends are what we're looking for so that we can then combine these sticky regions with new DNA, allowing us to produce a recombinant molecule. And then DNA ligase will come along and join the sugar phosphate backbones together. So remember in chapter 8, we talked about restriction enzymes and their use as protective, basically the immune system, one of the immune system structures of bacteria. They protect bacteria from foreign DNA, mainly viral DNA. So we now use these restriction enzymes so that we can modify the different bacteria and other organisms. So here are 3 types of restriction enzymes. Restriction enzymes are always named for the bacteria that make them. So ALU1 comes from Arthrobacter luteus. That's where ALU comes from. And then 1, this is one of the first ones that it makes, okay? So ALU1 cuts DNA at a section where you read forward AGCT and backwards AGCT. So palindromic. It cuts between the G and C both times. So this is a blunt end cut. Blunt end cuts are beneficial to the bacteria. They'll kill they'll break down the DNA. And that means that the DNA can't control, so viral DNA can't control the cell structures. But it's not useful in biotechnology. We want those sticky ends. So here is BAM-H1, Bacillus amyloliquefaciens H1, because again, it's the first one. Here, BAM-H1 cuts between the G in a palindrome that reads GGATCC. It cuts between the GG. So it cuts here, goes over, and cuts there. So you have this sticky region here. So this would be beneficial in running biotechnology or trying to produce a new recombinant DNA sequence. And then EcoR1 also cuts between a GA, so produces that sticky end. I'm going to stop here and I'll get into the next tool, which is gel electrophoresis, in our next video. Bye.